Hi everyone, my name is Beatriz Picinato and I am a PhD student at the University of São Paulo, Brazil. I will present the poster entitled Comparative Transcriptomics Among Archaea to Search for Circular RNAs. So circular RNAs are RNA molecules that have their 3' and 5' ends covalently ligated. Today, we know that circular RNAs are present in all three domains of life, and they are increasingly being understood as functional molecules and not as mere splicing byproducts. For example, circular RNAs have already been associated with cancer and in the immune system in mammals. However, despite being prevalent and conserved in eukaryotes, circular RNAs have not been consistently reported in prokaryotes. Only a few specific studies were done in bacteria, and only 2 transcriptome studies to search for all the circular RNAs of an organism were performed in archaea. So archaea are prokaryotic organisms with both bacterial and eukaryotic features that are well placed in the tree of life to study the evolutionary paths that established circular RNAs. So in this study, we aim to identify and characterize circular RNAs of Halobacterium selinarum, a model systems biology archaeon, and to investigate circular RNAs' evolutionary history and conservation, especially among archaea. So to achieve that, we generated Halobacterium selinarum RNA-seq data in which the RNA was pre-treated with RNase R, an enzyme that digests linear RNA and enriches for circular molecules. We are calling this type of sequencing circ-seq. We also gathered from the literature circ-seq data from other archaea for reanalysis: Haloferix vulcani, Paracoccus abyssi, Sulfolobus acidocaldarius, and Saccharolobus sulfatericus, which represent all the available archaeal circ-seq data. All archaeal datasets were aligned to their respective reference genome with an in-house established pipeline. The high-quality, non-aligned reads excluded by this pipeline that also survived RNase R digestion went through Monarch-seq, a pipeline we developed to search for circular RNA signatures in prokaryotic RNA-seq experiments. From all the circular RNAs we found, only those supported by 15 reads or more were considered for further analysis. So we could find circular RNAs associated with 4 major classes of transcripts in the 5 organisms: ribosomal RNAs, transfer RNAs, small and CD box RNAs, summarized in the sRNA category, and insertion sequences. So we could find circular RNAs associated with ribosomal and transfer RNAs that were already described in the literature for archaea. While in the other organisms the circular intermediates were already known, they were not properly demonstrated in the Halobacterium selinarum until now. So we could find these canonical circular ribosomal RNA intermediates, highlighted in purple in the panel A, and also the circular tryptophan intron that are present in all archaeas studied except for Paracoccus abyssi, as shown in the panel B. The processing and circularization of these RNAs occur in the Butch-Helix-Butch RNA motif. While circular RNAs associated with the ribosomal and transfer RNAs were already described in literature for archaea, we could also find a novel class of circular RNAs associated with a family of insertion sequences that are ancestral to the Cas proteins in Halobacterium selinarum and in Sulfolobus acidocaldarius. In Halobacterium selinarum, the circular RNAs are associated with a class of no-coding RNAs previously described by our research group, represented in purple in panel D. So we successfully developed Monarch-seq, a computational pipeline to search for circular RNAs in prokaryotic RNA-seq data that could retrieve previously known circular RNAs in archaea. And besides that, we could also detect novel circular RNAs associated with insertion sequences ancestral to the Cas proteins, opening the door for the better understanding of the function and regulation of these potential new genome editing tools. So with our reanalysis of archaeal circ-seq data and incorporation of a new dataset from Halobacterium selinarum, we observed that circular RNAs are conserved and abundant among archaea, maybe more than previously appreciated. So I would like to thank all lab members and collaborators that made this project possible, and give a special thanks to Vinícius, who made a major part of this analysis with me. I would also like to thank all the funding agencies that funded this work in some way. And thank you for listening.